Rajiv. I'm not sure where he's at the moment. It's a pleasure to be here. It's the first time for me to be here, to visit India actually, and I'm just coming from a very different meeting to the meeting we are here. I'm coming from a meeting of the local agricultural R&D community in Punjab, state of India, and I must say it was a very, very insightful, eye-opening and humbling meeting because researchers from the state of Punjab gathered to really address the real-world needs of farmers in the regions, and the same meeting was attended by thousands of farmers. It was really a very unique experience for myself, I must say, and I think it sort of helps myself, and I think should be an eye-opener to us to really keep focusing on the needs of the ultimate beneficiaries of the work, of our work. We're using all these new fancy technologies, but I think we cannot lose sight of what are really the needs of the ultimate beneficiaries and what we need to focus on. It was a very, very good meeting. Well, today I'm going to talk about a very large project that was initiated about 3 years ago at CIMMYT, funded by the Mexican government. And this is a project to go in and systematically characterize and mobilize genetic resources of maize and wheat. There are 2 very large gene banks at CIMMYT conserving maize and wheat genetic resources, very large numbers. In the case of maize, CIMMYT is holding 27,000 accessions, mostly land races. In the case of wheat, somewhere between 120 to 140,000 accessions, depending on who you ask and the day you ask. So far, we have been very good, as Andreas mentioned, in collecting and conserving these genetic resources, but I think we have not been, we have been lacking the tools and means to really go in and systematically unlock and harness the power of genetic resources in these gene banks. I'd like to start by acknowledging contributions of many, many people in this project, and this is only really a small fraction. Sarah Hearn is in charge of the maize component of the project. Alberto Romero has been doing some of the GbS analysis I'm going to present. Will Lee has also been contributing to the GbS analysis. Carolina Sansaloni, Cesar Petroli, Aleida, Hector, and Manuel have been generating tens of thousands of GbS data for the accessions. Martha Wilcox was involved. She's the phenotyping coordinator for maize. Sukhwinder Singh, who is sitting over there, is in charge of the wheat component of the project. Gary Artlin was involved in some of the original design of the maize GbS experiment. Andre Killian is here with us as well. Ed Buckler at Cornell University is a big collaborator on the maize side as well. So Seeds of Discovery is one of 4 components of a Mexico-funded initiative that ranges from characterization of genetic resources to their utilization in breeding programs and delivery of improved seeds to farmers through a component that focuses on seed systems, on delivery of seeds to farming communities and the associated agronomic practices that need to go along with improved seeds. So Seeds of Discovery in a way is sitting at the interface between genetic resources on the one hand and breeding programs on the other. Why this project? We really need to, until 2050, we need to bend this curve upwards, these 2 curves upwards to the targets that the FAO estimates in terms of what's required, what yields are required to feed humanity at that time. And it's not an easy task. And we believe that genetic resources in the face of climate change, soil degradation and falling water tables, increasing costs of energy and fertilizers, genetic erosion, I think genetic resources are really one of those few last remaining options that we have to continue raising yield in coming decades. The research project is a decidedly applied project. We are not going into genetic resources to characterize them because it's interesting to do so. It is interesting to do so, certainly, but that's not the primary motivation. We're really focusing on not only characterizing, but also mobilizing, actively mobilizing new genetic variation into breeding programs. We are less focusing on upstream research, although I think we're going to generate a lot of data that can be mined for more upstream research, but for that purpose, we'd like to collaborate with others rather than doing it ourselves. We are focusing mostly on genetically complex traits because those are the important traits: yield potential, heat and drought tolerance. Although on the way, we certainly want to harvest low-hanging fruits in terms of simple traits if we identify, for example, disease resistance traits that are caused by single genes. The project has overall 3 main components. One component is related to the systematic characterization of genetic resources. We are going to characterize using GbS the entire gene banks of CIMMYT and also other gene banks, particularly in Mexico in the case of Maize, because there are other gene banks in Mexico that have very valuable genetic resources not conserved at CIMMYT. Essentially, we want to create a molecular atlas for each of the 2 species. We want to identify sources of underutilized genetic variation. We're looking for selection imprints, both environmental selection imprints and also selection imprints caused by past breeding efforts, particularly on the wheat side. Because essentially the whole breeding history, wheat breeding history of CIMMYT is conserved in CIMMYT's wheat gene bank, and we can go back 30 years and look for selection imprints due to breeding. Also, in the case of maize, we want to look for potential heterotic pattern or patterns that could be implemented, or in the case of wheat, we will be looking for hidden translocations that I'm pretty sure we're going to find. Combining genotypic data generated under this component with phenotypic data generated here, we are going to perform a range of association mapping analysis. I'm going to show you some examples from the maize side in my talk today. But both of these components really only exist because they feed into what we call the development of bridging germplasm. It's essentially systematic introgression efforts, pre-breeding efforts to generate germplasm that is more breeder-ready than the original accessions in the gene banks, essentially trying to systematically express novel genetic variation in predominantly elite genetic backgrounds. These are it's a broader view of the project. I've mentioned the first 3 components. We also have 2 other components, one related to information management and software development. We realized that the project is generating a lot of data. We are not ready to manage all this data, so we are also investing in software development and capacity building efforts. And I'm going to talk now, this is all screwed up, I'm sorry, about the systematic diversity surveys, an example for GbS analysis in maize, and I'm briefly going to mention the capacities generated in Mexico in terms of establishing a genetic analysis service that actually processes all these samples. We decided at the beginning of the project 3 years ago that genotyping by sequencing would be the principal method of choice for genetic characterization efforts, essentially because we felt that really the transitioning from genotyping by assay methods towards genotyping by sequencing was a definite transition that is similar to what happened in the area of photography, from analogous to digital photography. Once you start using digital photography, you don't turn back. And it really creates data sets that are forward compatible. The sequences, even if in the case of wheat, we don't yet have a reference genome, the GbS tags are ready to be aligned against the reference genome as soon as it becomes available. Other speakers have already mentioned other reasons. We essentially can avoid ascertainment bias. We simultaneously discover new genetic variation and score or classify the allelic states of these newly discovered markers. It's a configurable platform, and we're using 2 different systems, or 2 different flavors. One is the DartSeq platform, which uses 2 as opposed to 1 enzyme. We usually get around 60 to 7,000 markers. That's the sum of both SMP markers and presence-absence variation markers, which, as Andre pointed out, are really is not a very good term, because presence-absence really refers to the genome representation, but not the underlying genomic sequence, because they can also be caused by SNP variation in restriction enzyme sites. So but in any case, there is a lot of information generated, certainly sufficient for an initial diversity assay. We usually obtain about 20 to 35% missing data. The 20% usually in wheat, 35% usually in maize, because there's more true presence-absent variation in maize probably than in wheat, at least in the materials we're looking at. We usually have somewhat lower error rates, and we can call heterozygotes for a subset of the GbS tags, and we don't use imputation for those data sets. And we use that to build the molecular atlases. We use the Cornell method, which maximizes the number of markers being scored simultaneously specifically for wheat association mapping, because there we are constrained by extremely little linkage test equilibrium. So we need to maximize marker density. There are, of course, more missing data, and we need to use imputation, but we can increase marker density. We have established, in partnership with Diversity Race Technology, a genetic analysis service in Mexico. That was one of the ways to kill two birds with a stone in terms of creating local capacity and economies of scale to be able to process these enormous numbers of DNA samples, while at the same time having a vehicle to build capacities within Mexico and the region, and have a service available to support local agricultural research, particularly applied agricultural research. The vision is to not only provide DNA to raw data service, but also work in the area of converting data into knowledge with future clients, and also deliver services related to information management, informatics platforms, etc., to support breeding programs. And I also mentioned briefly, we're working on software development as well. Briefly, we're working on the KDDART database, implementing KDDART database, and try to link that up with other platforms that either already exist, like Green Global for managing genetic resources, or a web portal developed by James Hutton Institute in the UK, ready to deliver data over the web. In the case of wheat, where we stand at the moment, we have completed the characterization of about 42,000 gene bank accessions already. So each of them has been characterized using GbS. We selected only one individual per accession. After some initial surveys showed that there was relatively little genetic variation within the seed bags, so to speak, within the accessions. Although we also clearly detected cases of sample mix-ups where we had accessions or bottles of seeds that were clearly mixtures of different accessions or varieties. So it's never, I think once one goes into characterizing systematically gene bank accessions, a lot of skeletons will be unearthed, I can tell you. I've seen a few. As I mentioned before, we obtained about 60 to 65,000 data points per sample. We are at the moment processing all this data for comprehensive diversity analysis and the design of association mapping panels. We will collaborate with DART, with the consensus map that you have built for weeds to order these markers along the genome, and the overall target will be somewhere around 160,000 accessions, not all from CIMMYT. Just one example of a little core collection or association mapping panel that we built from Iranian land races, 2,400 Iranian land races in CIMMYT's gene bank, and we built a little subset that is going to be used for association mapping. It's not only that we are genotyping, but we also, on the weed side, we have been systematically phenotyping quite a large number of accessions. For example, about 70,000 accessions have already been screened in the field for heat and drought tolerance. So we are combining those data with the genotypic data to build association mapping panels where we try to spread as much as we can across the available genetic diversity to cover the genetic space, at the same time trying to enrich for good phenotypic values in terms of heat and drought tolerance, to enrich the association mapping panel to some degree at least with positive alleles for heat and drought. In the case of maize, we have a very different situation, because in maize, well, first of all, I think 25,000 of the 27,000 accessions in the Maize gene bank are land races, so they're all highly heterozygotes. And we need a different approach. We need, and that's one of the reasons why we're using the DART-seq approach, because of its ability to score heterozygotes for a subset, at least, of the GbS tags. The good thing is the system is based on PSD1 enzyme, which has a partly overlapping restriction enzyme recognition sequence with APEK1, which is the enzyme that Cornell University uses. So if you apply both techniques on the same samples, you have some GbS tags that overlap. But that's only one problem. The other problem really is because the accessions, Maize accessions are land races, each accession contains a lot of genetic heterogeneity within the bottle of seeds that is being used to conserve it. So in theory, we need to characterize not only one seed per accession, but several seeds, and it would be basically too expensive if we went in and characterized individual seeds in each of the accessions. So Sarah Hearn worked with Andre Killian to develop a method to apply GbS to pools of DNA, and that turned out to work really well. What you can see here is a comparison of allele frequencies evaluated by genotyping the individuals within a particular accession separately, and by genotyping a bulk of DNA representing 30 individuals. And there's a reasonable correlation between the two, which means that for at least initially for the first pass characterization, we can use DNA pools representing Maize gene bank accessions and obtain both information on allele frequencies within accessions, but also genetic distances amongst different accessions. And for that, we use mostly DART or in silico DART or presence-absence variation markers, polymorphism, which allow us to establish genetic distances between the DNA pools, so to speak. Here's just an example of the initial experiment that we used to optimize the methodology, where we compared separately assembled bulks of increasing size. And as you can see here in colors, once you're above about 32, there's little change. So we used 30 leaf discs bulked from 30 individuals and extracted one DNA sample from these 30 leaf discs after showing that if you pool at the leaf disc sample level or the DNA sample level, it doesn't really matter. You obtain very similar results. And this method has really allowed us to move ahead very quickly and essentially bring down costs by an order of magnitude in terms of characterizing the whole gene bank accession. So what we're doing here is, for each of the accessions, we sample 30 plants each and subject that to a GbS analysis to determine allele frequencies within accessions and genetic relationships amongst accessions for a range of different applications. And we just, by the end of last year, we finished sequencing the first 20,000 Maize accessions from CIMMYT's gene bank using this methodology. In total, we identified about 230,000 SNPs in 230,000 in a slightly smaller number of GbS tags. Although I think a reanalysis of all the 20,000 together is probably going to increase this number to some degree, but will be interesting to see. Interesting is also only 20% of these 230,000 map to the Maize reference B73 genome, which is not surprising, although the extent of lack of overlap is perhaps a little bit surprising. The reference genome was constructed from a temperate Maize line, and here we're dealing with tropical and subtropical materials predominantly, and there's a lot of fluidity in the Maize genome. So we're also working together with an institute in Mexico called Langebio to whole genome resequence a set of about 20 land races to generate a sort of tropicalized Maize reference genome to be able to better align the GbS tags against a whole genome sequence. Clearly, the GbS tags are enriched for gene-rich regions. Here's a distribution of the number of SNPs across one of the chromosomes, and you can see here At the centromere, there are less markers, and in more gene-rich regions, in more distal areas, you have higher frequencies of numbers. And our target is about 40,000 accessions between CIMMYT's gene bank and several of the Mexican gene banks. Next steps, we will be looking at environmental selection imprints. Most of the gene bank accessions have fairly good GIS data associated with them, or geographic data associated with them. Essentially, we would like to identify allele frequency gradients across environmental clients. For example, we want to look also at breeding selection imprints. We have several examples at CIMMYT where we have materials that have undergone recurrent selection, several cycles of recurrent selection. We can look at how allele frequencies across the genome have changed over time. We also will be looking at race-specific footprints. Now, finally, to finish off, I'd like to give you an example of the GbS, a very large GbS experiment that we did also on the Maize side. The design was a bit unique, I would say. We started with an existing core collection of about 4,500 accessions that predated the project. From each of the accessions, we took a single individual and crossed it with a tester adapted to one of the 3 geographical regions of maize production in Mexico, the tropical one, subtropical one, and highland. And the rationale behind this approach was an expectation that haplotypes would be replicated across different accessions. So we wanted to measure haplotype effects, and we thought that it was better to sample broadly across many accessions, provided that haplotypes would be replicated, than to go and characterize and sample deeply in each of the individual accessions. The testcross progenies were then phenotyped in field trials across Mexico, and the testcross parents were subjected to GbS analysis, both the Cornell flavor and also Darzig at the same time. A lot of phenotyping work during 2.5 years was done by not only by CIMMYT, but also by many collaborators in Mexico, mostly by INIFAB. INIFAB is the USDA equivalent in Mexico. And a total of 700,000 individual data points were collected in 34 trials across 14 locations, collecting traits that were originally prioritized by a group of breeders. And as I mentioned, the testcross parents were genotyped with the Cornell University GbS approach to maximize marker density, and with Darzig to identify areas of heterozygosity in the genome at the same time. And the missing data was imputed not only using data from the Seeds of Discovery project, but essentially everything that had gone through the Cornell pipeline, GbS pipeline for maize. Luckily, there was little structure in the core collection that pre-existed the project. And we first looked at flowering, male and female flowering, and just to make sure that we identify those regions and genes that we expected we should identify. And indeed, that was the case. Well, one of the first conclusions was that the marker density was just sufficient. As you can see here, for example, there are 2 genes identified that are known to be in the pathway of female flowering. The marker density in this region, or in general, once you zoom in, is not very high, but still we were able to identify some of these genes. In fact, quite a few of the genes involved in female flowering. In the case of male flowering, we detected an inversion that came originally from Teocinte, the progenitor of maize, and which turned out to be very prevalent in highland materials, and that was actually the strongest contributor to controlling male flowering. Here's another example of a disease, a very important disease, emerging disease in the southern part of Mexico. It's called tar spot. We identified, excuse me, 2 accessions that were really quite tolerant or resistant to the disease. the test crosses still were quite resistant, and luckily we got one marker here on chromosome 9 that seems to be associated with resistance to this disease, and when you zoom in, you actually find a disease-resistant gene right next to it. So the conclusion here essentially is these are first-stage analyses that inform us how much more phenotyping do we need to do, and how much more would we perhaps need to increase marker density to make sure that we can actually pick up most of the signals. Because if we identify one locus that clearly contributes to disease resistance in this case with a single marker, how many else we may have missed, we don't know. Finally, I'd just like to highlight before finishing, again, these two components exist because they feed into the third component, the development of bridging germplasm, based on introgressing new genetic variation into elite germplasm selected by colleagues at CIMMYT, both on the maize and wheat side, and in many cases trying to use new approaches and technologies such as genomic selection, particularly for mobilizing genetically complex traits. The idea is here to develop breeder-ready lines and populations that are useful both for genetic analysis purposes, such as family-based association mapping, but also serve as a starting point for trait mobilization into breeding programs, and deliver those populations and lines alongside the genetic knowledge attached in terms of molecular markers or genomic prediction values that inform breeders or additional tools to breeders to aid the mobilization of this genetic variation into breeding programs. And I think I skip over the next two slides and finish by acknowledging a very large group of people who recently met end of last year. These are all people participating in the project. You can see there are many collaborators from Mexican institutions, also participants from other countries, and I just realized that I forgot to mention John Hickey, who is sitting, where is he? Yeah. John? Here. Who is one of our collaborators as well, who is here with us today, working on simulations of pre-breeding strategy for maize using genomic selection. It's a very big group of people. It's a very excited group of people. Every time we get together, there are lots of interesting discussions and new ideas that are being floated. It's a very rewarding project, and I think it's a project whose time has come. And returning to what I said at the beginning, I think it's a project that needs to stay focused on what it wants to deliver and why it exists, and that's to accelerate breeding through the mobilization of new genetic resources, to broaden the genetic base of breeding programs, to accelerate genetic gains. Thank you very much.
Thank you, Peter. First of all, let me congratulate Peter and his large group of people doing this tremendous work. The work is very important for crop improvement because large genetic resources are available, and it's good to see that the new technology is moving into that direction. With that, I would like to take some questions from you, please. Roberto first.
Does it work? Okay.
Yeah.
I'm not sure if I saw it well. I think your peak was on VGT1 for flowering time. Well, the reason I'm asking, because I was a bit puzzled by the fact that you have photoperiod sensitive material there, right? So, because that was the largest peak you detect for VGT1? Is that VGT1 or not? I cannot read.
That's probably...
Looks like on chromosome 8.
I think it's VGT1, yes.
Okay.
I think so.
So in a way, I'm very pleased because, you know, the reason we cloned VGT1 because we knew it had a large effect, but I'm pleasantly surprised to see how big the effect is in a background where you also have photoperiod sensitive genes.
Yeah.
And my other comment is the other peak that you picked up, are they photoperiod sensitivity or flowering per se?
I don't know, I'm sorry.
Okay.
This is I think I can forward the question to Sarah Hearn, who is in charge of maize.
Right.
And if you want, I'll do that and I get back to with you with an answer.
No, I mean, it's just curiosity because
Yeah.
I worked on VGT1, and usually when you put VGT1 in a context where you have photoperiod sensitivity, it doesn't stand up as much as it does. So in a way, I'm very pleased to see how important that locus is.
Yeah, I mean, I'm not sure how the size of this peak compares to other, to other peaks that were detected. Again, something that I can gather from Sarah and forward to you.
Well, but if you have female flowering time, it seems the largest QTL there, right?
Well, within this window, no, that's not the whole genome scan. That's just.
Oh, I'm sorry. I thought it was.
That's zoomed into a small region within one chromosome.
I apologize. I thought it was the whole genome scan.
No, that's not. We have a little bit more than a few dozens of markers. Yeah.
Oh, okay,
so
okay.
That's just a small region. No, that's
but
a small region.
Maybe I missed that, and I do apologize.
So there may be other peaks that are higher.
That what I would have expected. Yeah. Okay, thank you. Sorry.
No worries.
There's one more question.
I think so.
Peter, you mentioned the private SNP alleles that showed a correlation to some of your traits in GbS, right?
Which allele? Sorry.
They were private. They were singular SNP alleles that were associated. Can you just give us an idea on the LD in that population and the marker coverage that you had in your GbS?
Here we had about 800,000 SNPs called across 4,500 or 4,300 or so testcross parents. The LD, again, I would need to ask Sarah to get that information. But I know that it was very, very small and smaller than what you usually see with temperate materials. Because, I mean, that's the center of diversity, center of origin of maize, and these are ancient land races, essentially. There's very, very little LD. So our greatest fear was that even 800,000 markers would not be sufficient to robustly detect genome regions using GWAS. And I think we detected some that we expected, but it's likely that we did not detect all that we could detect had the multiplexing level been even higher.
Okay, with that, please join me in thanking Peter and his colleagues for the excellent work. Thank you.